Hello and welcome to the Onion IQ podcast. I'm Alice Formiga of eOrganic, and today I'm talking with Dr. Brian Kvitko of the University of Georgia about the biology of two onion bacterial diseases, Burkholderia gladioli and Burkholderia cepacia. This podcast is brought to you by the USDA NIFA OREI funded project Onion IQ, improving quality, resilience, soil health, and food safety in organic onion in the southeastern and southwestern United States, led by Dr. Babesh Dutta, also of the University of Georgia. You can find out more about this project by going to the website eorganic.info/onion. So today I'd like to introduce Brian Kvitko, who is an associate professor in the Department of Plant Pathology. He specializes in the use of genetics to understand interactions between plants and bacteria. He's going to give a short presentation, and then I'm going to ask him some questions about how to manage these diseases on organic farms.
Hey, thanks so much, Alice. Yeah, I'm happy to be here. So let me tell you a little bit about these problematic bacterial pathogens that we're dealing with in onion. In particular, the focus of this group is the Burkholderia onion pathogens, of which there are many, but we're focusing on two in particular: Burkholderia gladioli. I'll usually just say B gladioli because it's quicker to say, and Burkholderia cepacia. Okay, so Burkholderia bacteria. Um, like most bacteria, there's not really that much to look at. First of all, they're nearly impossible to see without a very good microscope, but even then, you're not going to have much to look at. Your average bacteria is about 100 times smaller than a human hair. So if you imagine taking a hair on your head or your arm and cutting it in half like a tree trunk, across that trunk, you could line up 100 bacteria end to end. They would fit comfortably there. In terms of their shape, they're sort of a rounded rod. They look a little bit like a gel cap, but obviously in miniature. Burkholderia bacteria are, we call them gram negative. They basically have two different cell membranes sort of sandwiched together, which does make them resistant to a lot of antibiotics. This is the same type of bacteria family, more or less. It's a big family, but for instance, you may have heard of E. coli or Salmonella. They're different than like the things that give you strep throat. This is sort of down in the weeds, but it's worth mentioning. They get around their environment. They have these little spinning flagella tails that they use to move, and they actually propeller themselves through their environment. Um, so in terms of what they look like, again, not that much to see. You wouldn't even necessarily know under the microscope that you were looking at Burkholderia. So the secret really for these organisms is in their genes. So Burkholderia as a group, they've got these unusually large genomes for bacteria. They're actually have multiple chromosomes, which is also a little atypical. So about 8 to 9 million bases of DNA. That's almost twice as big as your average bacteria. Usually, a bacteria will only have one chromosome and about 4 to 5 million bases. So pretty chunky on the genetic side. Just for comparison, though, even though it's big for bacteria, it's still very, very small compared to you, for instance. You have 3 billion bases. But just so you don't get too much of an inflated head, onions actually have 16 billion bases. So onions have humans beat in terms of their size of their genomes as well. So Burkholderia are sort of characterized by having very high metabolic flexibility, so they can use all different types of things as food. They can break down lots of different compounds to sort of provide for their basic nutritional needs. Um, Another thing that sort of characterizes them is that they have this high degree of drug resistance through efficient efflux pumps. So what that means is that they can take drugs and just very effectively and rapidly sort of pump them out of the cells. And that means that like a lot of antibiotics that we would use that would kill other things, they can just tolerate massive doses of antibiotics without really slowing them down. They're really tough little bugs. Now, many Burkholderia species are just common soil residents. In a normal, healthy soil, you would have Burkholderia present. Soil is a really high competition environment for bacteria. It's actually one of the most diverse ecosystems we're aware of, most diverse microbial ecosystems. Now, how do they survive in this very challenging, high competition environment? Well, some of those features I mentioned, like being very resistant to antibiotics, like being very metabolically flexible. Um, But they also produce a diverse array of natural products. Now, what are those? Those are things like antibiotics that help them sort of compete with other microbes in the soil. But unfortunately, a lot of those compounds also can help them interact with not just fungi, not with bacteria, but also with their host plants or with us. So there actually are some Burkholderia that are cause food poisoning in humans. Now, again, most Burkholderia harmless, harmless to humans, harmless to plants, just normal native flora of the soil. And some are even beneficial. There are Burkholderia that are very effective for biocontrol. There are Burkholderia that can be good for growth promotion, or there are even Burkholderia that participate in nitrogen fixation in sort of nodules with certain types of plants. But some are opportunistic pathogens of plants or even people. The pathogens that we're going to talk about today, or the ones that we're particularly focused on in this project, are Burkholderia cepacia, and that actually takes its name from onion, Allium cepa, so cepacia, of onion. And it's been known to be an onion pathogen since the 1950s. It causes a disease called sour skin on onion bulbs, and yeah, I think you can probably get some sense of why it's called that. It makes the onions smell kind of acidic and vinegary, and it smells bad. And you can occasionally get symptoms caused by this pathogen on the onion leaves as well as the bulbs. We also are looking at Burkholderia gladioli. This is sort of in a different, you can kind of split the Burkholderia family tree into three big groups. This is actually from a different group of Burkholderia. Burkholderia gladioli also can cause disease on gladiolus bulbs, which are these very pretty ornamentals. It can cause on mushrooms. And this is one of these ones that has been associated with food poisoning on coconut tempeh. And it causes a different disease called slippery skin. The reason it's called slippery skin is if you kind of look, this is a very dramatic example of this disease. But basically, it rots out the inside to where if you were to pull the stem of the onion, the whole center of the plant would just come out and would leave the bulb behind. It's slippery. It slips out of the sort of infected onion. And this pathogen does routinely cause symptoms on leaves. We can see some of those leaf symptoms here. Okay, so one of the questions, actually, even though we've known about these as pathogens for, in the case of Burkholderia cepacia, almost 70 years, it's been sort of a challenging question to understand how they cause disease. So this is a little cell diagram of sort of what we understand about how these different organisms infect their host. So B. cepacia secretes enzymes that actually seem to be involved in degrading the onion tissue. So things that can break the onion cell wall down. So kind of like it's not that extreme, but I'm sure you've probably heard about flesh-eating bacteria. So like that, but if it was an onion. So um gladioli also secretes some of those enzymes out, but it produces one of these natural products called toxaflavin, which actually is a yellow colored compound that is important for it to be able to cause disease. So they kind of they're similar, they're related, but they do cause disease in different ways. Another thing that we did notice, though, is that for the strains, for the versions of Burkholderia that are adapted to onion, we also identified that they carry genes that allow them to be resistant to the sort of sulfur compounds that onions produce when they're damaged. So this was sort of a key feature that we picked up on that they both had, even though they're from these very different groups of the Burkholderia family tree. Okay. So if you've tasted an onion, presumably you have, I think almost everybody has, or if you've ever cooked with garlic, you know that onions can make your eyes water. So why is that? Essentially, the onion bulb is how onions reproduce themselves, and they want to defend that structure, and they do that basically by producing a form of tear gas. When the onion tissue is damaged, you will be mixing two things together. You mix an enzyme and a precursor compound, a chemical, and that will reduce this lacrimatory factor that is released. It gets volatilized. It interacts with the membranes in your eyes with water, and produces a very dilute sulfuric acid. Basically, it makes you cry. That same family of chemistry, though, is actually involved in what gives onions their flavor. So the flavor you would associate with onion or garlic comes from this same type of chemistry. And that same type of chemistry also has antimicrobial action. So basically, the onions are protecting themselves when they're damaged against herbivores that would want to chew them up or microbes that would want to sort of colonize their tissue. So, what we learned is remember, I mentioned we said that we had found those genes in both of these types of pathogens. We had studied and found similar genes before in other bacterial onion pathogens. We saw that the Burkholderia both had them, that they were similar to the ones that we knew about. We also learned that if we went in and we clipped those genes out of the pathogen and studied them in the laboratory, that basically, if we took these genes away, they didn't cause disease nearly as effectively. So we knew that this was an important factor for them as well. So we call these the thiosulfonate tolerance genes. And what this is maybe a little hard to see, but you can see here what we've got is an onion leaf, an onion blade. You can see that when we have a strain where we've clipped these genes out, you really don't get any symptoms. And that's something that we've seen from multiple different strains at this point. So what we've been doing is because basically using these genes as a way of tracking how much Burkholderia we've got in soil. We're using DNA extracted from soil to survey how much of our onion pathogens are present out in that soil. So how do we do that? We get samples of soil from the different fields that we're exposing to different treatments. We can actually purify total DNA from that soil. So we break down, all of the bacteria, all the microbes, they all get broken apart. We purify the DNA, just the DNA away from that soil. And then we can do a molecular assay where we can count how many copies of those thiosulfonate tolerance genes were present in the original soil sample. And this is a very rapid and accurate way to figure out how much Burkholderia is present. It's actually surprisingly, remember, like we're talking about things that are a hundredth the size of a human hair in soil. It's very hard to accurately get an estimate of their population. So this lets us sort of get to that question in a very quick, rapid, and effective manner. Okay, so now we come to the question of like, okay, well, how do we manage these diseases? So in plant pathology, we have the concept of the disease triangle. We know that we need three things for disease to occur. You might remember this as like the fire triangle or something like that. So what do we need to have to get disease? Well, we have to have a susceptible host, we have to have a virulent bacteria, and then we have to have the appropriate environment. Now, if we want to look at managing these diseases, we want to manage one of these vertices, one of these angles. So, unfortunately, while there's some evidence of host resistance and the onion, we don't have great evidence that we've got really strong resistance out there in the onions that are available. And it infects through wounds and damp soil. So, it's also hard to control the environment. You can do it to some degree, but for the most part, you're kind of at the whims of whatever the weather brings your way. And again, most of the strains of Burkholderia are virulent. So really, the one part that we're hoping that we can really manipulate here is the pathogen. If we can reduce how much of the pathogen is present, that is probably the most straightforward way for us to manage these diseases. Just reduce the amount of pathogen that's available, because the other things are unfortunately harder for us to control. And the way that we're doing that, or the treatments that we're testing for the Onion IQ project, are two organic compatible treatments. One is soil solarization, which you may be familiar with, where the soil is heated up to kill microbes. Often this is done with large sheets of plastic to capture solar radiation. We're also using biofumigation. So you may be familiar with fumigation, where you'll take chemical fumigants and pump them into the soil to kill microbes that are present. Obviously, that's not going to be organic compliant. So instead, we use biofumigation. So we use special breeds of mustard that have been bred to be sort of very hot, like again, that the mustardy chemicals, the isothiocyanates that sort of give mustard its pungent flavor. can be noxious in high concentrations. So these special mustards have been bred to have a very high concentration of those compounds. So those are grown until they're flowering and then tilled into the soil. So essentially, we're doing something similar, but we're doing it in an organic, compatible way. And then we use our assay to figure out, okay, are we actually affecting the population in the Burkholderia in soil or not? Like, are our treatments effective? And if you want to learn more about this project and the work that we're doing, I'll direct you to the eOrganic site where you can see some of the work that we're doing, some of the people that are there, and some of our results. Okay, so I think that kind of brings me to the end of my presentation. Alice, you said you had some questions for me.
I do. And also, I just wanted to mention that you can find our website at eorganic.info/onion. Okay, so how do the infection pathways um, of these two different bacteria differ?
That's a good question. And honestly, I would say that this is something we're still trying to learn. It does look like both of them require wounds to enter. Most bacteria aren't able to sort of independently breach plant tissues. They usually need to access either natural openings or they'll take advantage of wounds that are created by arthropods or other things that are living in the soil. We also think that we may get rain splatter from the soil that will help to spread the bacteria up onto the leaves, which is how we get some of those leaf symptoms. But even then, there's this idea that on their own, the onions can kind of protect themselves until sort of a something has breached the surface and allowed them entry.
Okay. So does it affect all the onions in a field, or does it just affect like the ones that have some sort of a wound or opening?
No, it's only going to affect the ones that have experienced some kind of a wound. Like even if they were to spread from, obviously, if you get inoculum building up, if you get infection building up, you could imagine things like weather or rain allowing them to spread from onion to onion. But it's kind of in the same way that, like, you would need to have like, if you have an open cut, it can get infected, right? It's comparable to that.
Yeah.
Yeah.
Okay, great. So what symptoms should growers look for when they're trying to identify infections caused by Burkholderia gladioli versus cepacia? I can't pronounce
No worries.
cepacia in the field or during storage.
So I think for cepacia, it can be a little difficult because we usually think of it as primarily a bulb pathogen, a below ground pathogen. Um, I have colleagues who work in extension, and they would say one of the best ways to tell is actually the smell for
Mm,
cepacia. That vinegary, that sour skin odor can be a giveaway. For gladioli, again, if you've got it in the field and if it's severe, when you try to harvest the onion, you're not going to get a bulb. You're just going to pull the center right out of it.
mm.
You also can get those foliar symptoms. It's more common with gladioli, so you may notice it there. Obviously, the post-harvest is the worst situation where you've already harvested your crop, you've spent all that energy and everything, and then the disease doesn't occur until all of that energy, effort, and time has already been spent. Um, so I mean, the best thing is just, try to not get it to begin with, as you unfortunately.
So, are there any cultivars that are more or less resistant to these bacteria?
So I think one of the things to keep in mind is that gladioli and cepacia are going to be different. So you probably are not going to have one that's, see, it would kind of be like saying, oh, having good resistance to like every virus. It's like we need different vaccines for different things. Um, it's an important question, and I don't know any of the cultivars that are. I think there are some trials that have indicated that there's some degree of tolerance for some of them, but from a diagnostic perspective, this is why it would be important to know who is your big and major player. Do you have cepacia or do you have gladioli? Otherwise, you might be putting out something resistant to the wrong pathogen.
Yeah, okay, that's great. Um, so, are there any environmental conditions, you mentioned this a little bit, such as
Mhm.
humidity, temperature, or soil composition that make onions more or less vulnerable to these diseases?
Hmm. Let's see. I think again, like most plant pathogens, water, standing water, or how damp it is obviously a key thing. The presence of anything that would be below ground insects or things that could create entry points would be part of it as well. Burkholderia are pretty comfortable with warm temperatures. Actually, you can see sort of a seasonal march of different pathogens as the season progresses. So a lot of your Burkholderia diseases are more sort of a late spring, summer disease. So clearly climate is playing a role. In terms of soil composition, unfortunately, I don't know if we really know a good answer to that question at this point. It's really hard to study soil microbes.
Yeah, you mentioned there's so much going on in there and so much
It's it's
competition.
it's
So,
incredibly complicated, and I hate to say it's a bit of a black box. It's really difficult to get a detailed picture about what they're doing in there.
okay. How do these pathogens, well, other than the through injury entry
Mm,
points, how do these pathogens spread within agricultural environments? And is one species known to spread more aggressively or persist longer
mhm.
than the other?
So I think it's also kind of good to think about and realize that like these things, we think of them as pathogens, right? That's where we're concerned about them. Most of them will just hang out in the soil. They don't care. They're just going to take the opportunities that come by. They're very good at living in soil. One thing that we do have going for us is that soil pathogens typically don't spread large distances, at least under normal circumstances. They kind of you can get a lot of local variation. They tend to stay because soil doesn't move a long way, except humans do tend to move things for the pathogens, right? So I'd say the thing that really can allow things to spread is humans moving them around, right? Which to some degree is inevitable. We're interested in that question about which one lives in soil better. I think, again, that's something we're actively looking at right now. Cepacia seems to be very common in soil. We have found gladioli more sporadically, but that could just be our regional diversity difference. I don't know if that's a trend that's going to hold across like the whole country, for instance.
Okay, great. I have one more question,
Hmm.
and that is, what is known about the ability of these bacteria to infect crops beyond onions? And should growers be
Hmm.
concerned about cross-crop transmission?
Let me think. Cepacia is really known as an onion pathogen specifically. And I think we mentioned about how gladioli has some capacity to infect a wider diversity. Now, in plant pathology, we often split up pathogens into something we call pathovars, pathovariants. So we'll see that in some groups, for instance, it's the same species, but you've got one that infects tomato really well and another one that infects cucumber really well. So gladioli has been split up. Like there's a pathovar allicola, there's a pathovar gladioli, and all that stuff. So you'd think, okay, well that means I only need to worry about the allicola, right? Well, actually, no. It's a terrible system, actually, because if you take any of them, they kind of can infect any of them more or less. Um, I would say that we're probably not growing gladiolus or a mushroom in the same way or conditions that we're growing onion. So from that perspective, probably not. I did want to say though, as scary as it is, some of these are also known to be human pathogens under specific circumstances. So Burkholderia cepacia, for instance, can be a problem for people with cystic fibrosis. Can
Mm,
colonize their lungs, but that is a specific population that sort of has a particular, I don't know. I guess maybe it sounds mean to say immune deficiency, but let's say a comorbidity. Um, you shouldn't have to worry too much about these pathogens moving to other crops in general. Yeah.
like, how about other crops in the allium family or the lily family? Like, for example,
Oh, I get you.
garlic or leeks or, um,
Um,
I don't know, flowers in the lily family, things like that.
Flowers in the, I think gladiolus actually is in that family. I mean, I could be wrong. I'm pretty sure gladiolus is in that family. Garlic, there have been reported cases of Burkholderia gladioli causing disease on garlic. We don't hear about bacterial diseases of garlic nearly as much. They produce way more of those defensive compounds in general, but
Hmm.
it's not unheard of. And a lot of the things that can infect onion also can infect garlic under the right circumstances.
Okay, that's great. So I just want to thank you, Brian. That was great. It was interesting, and I learn more every single time we do this. So thank you very much for joining us today.
Yeah, thank you for the invitation. It was a lot of fun.
We will be posting more episodes of the Onion IQ podcast. So if you'd like to be notified about that, please feel free to subscribe to the eOrganic YouTube channel. And as I mentioned, you can find the Onion IQ website at eorganic.info/onion.